Thank you for inviting me here today. I'm not going to use slides. I'm just going to talk to you, and I have a few notes here that I'm going to look at every now and again to remind myself. So I've been asked today to introduce the concept of gene therapy, and I would seek forgiveness for those of you that know what it is and have a huge amount of experience, but bear with me in that some people in the audience don't really know what it is and don't understand why it's needed, and they don't understand why some of these quite new treatments, even some that are still under development, target a gene, or more specifically the DNA. That's the letters that code for a gene. So they often say, well, why is it necessary to go right back to the DNA? Because for scientists that is an unenviable task, and it makes the whole development process costly and also takes a longer length of time. So if we think of ourselves as a book, I think this is a good way to try and understand that the letters are the DNA within us, and then the words make genes, the sentences make proteins, the pages make cells, each chapter going on to make an organ, where the full book then makes us, our body, and what we are. So this entire book depends on the letters of the DNA being cracked from the very start. And that's where the problem lies with all of these diseases we're talking about today. So each of us has around about 3 billion pairs of these letters of DNA that encode us. And that's what makes us tall, dark, handsome, blue-eyed, smart, and all the rest. That sounds a bit like my children putting out an advert for a man for me. And truth be told, when they did try to do that, they ended up saying, "Mum," and this is a quote, "could you just cope if he just had his own teeth?" So trust me, your DNA is what makes each of you individual and each of you special. So I want you to think about, if you were to try and write your book, you would have to type 60 words per minute, 8 hours a day, for 50 years. So you're each a pretty big book, and that's what we are trying to deal with. The scientists are trying to deal with that. So I think it's important to keep that in mind, the complexity of you and what makes you up. So I'm often asked, well, how do these letters or these genes cause disease? So the genes we get from our parents are copies, and they work in exactly the same way as they do in our parents. And sometimes a gene is not perfect, and it carries a spelling mistake, and that's a mutation. All of us have some of those mutations, and they're not always harmful, but in some stages they are, and some individuals and some certain mutations in certain genes, they do cause a genetic disorder. And unfortunately, out of about the 20,000 genes that we each have, it only takes one letter to be wrong in one gene, and that will cause the disease. And in the eye, and specifically the retina, there's quite a few of those inherited diseases that are caused by just a single gene mutation. So that's just that one letter that will go on to cause that disorder. I think it's important to think about, how do we know which letter in which gene is causing a disease? And I think there's a holding slide, if we have it up. There's a slide that shows a needle in a haystack. And the reason for that is that I've been there, I've done that, and I've looked for that needle in that haystack, and I've found that one letter and that one mistake that causes a severe form of genetic eye disease. It's very easy to pick up a book, to open it, to read it, but can you imagine trying to read your own book and the DNA letters and find exactly which one is causing the mistake? And I think it's unbelievably remarkable how we as scientists today can use sequencing technology, and just by using a sample of cells or a sample of blood from an individual, we can access their DNA, and we can find out the letter out of all those billions of letters that are causing the disease they have, be that an eye disease or any other disease. And I think today we take that for granted, and that you're able to go in and give a sample and ask for your DNA sequencing to be performed, and you want to know what's causing my disease, what's the reason for this genetic disorder. But you need to remind yourselves that it took 23 years to sequence the first human genome. Today, you could probably get that done in a matter of, well, a day. So a tribute to that and to the scientists involved in that allow therapies, because we cannot do that without that capability. And it is that ability to read our DNA letters, to find an error, and to diagnose a genetic disease that allows a clinician or a clinical team to then inform you as a patient, or any patient, and direct them towards the relevant treatments and or clinical trials specifically targeting that error that they have. And it's that diagnostic power that is the critical first step in what we would refer to as a successful sort of partner diagnostic therapeutic approach. And it's key to remember that everything is the crux of the diagnostics to start with. So as you already mentioned, Kate, there's been a discovery of, over the last 35 years, of over 300 different genes responsible for all the different inherited retinal disease and affecting millions of people worldwide. And gene therapy, which is quite simply fixing that mistake, is all about fixing that one letter in the book of life of any one individual. For me, gene therapy sits within that era of, you'll hear things like personalized medicine, precision medicine, preventative medicine. And it's really important now to understand that the therapies that we have now are for illnesses that we previously would have said were just untreatable. We would have labelled them as untreatable. So the eye, and the reason I think a lot of the eye therapies are the first to debut and reach the market, and we hear a lot more about eye gene therapies. The eye is an ideal organ for gene therapy because of a few unique features of it, but also its small size and its location. So it's quite easy to access it, making it quite easy to treat. We're not trying to do an intravenous injection and treat organs deep within the body. And it's also easy to visualize and view and examine and assess if a treatment is effective using all the different imaging and functional tests that we have available to us. So there are different types of gene therapies, and I've been asked to sort of touch on gene augmentation, gene replacement versus gene editing versus gene silencing versus gene knockdown. They're all gene therapies. But if we think of some of the inherited diseases of the retina, they require 2 copies of a bad gene, one from mum and one from dad. So because those both copies are bad, they do not have the gene that is responsible for making a certain component or a certain element in their eye. And the only type of gene therapy that's suitable for that would be gene replacement or gene augmentation. And that's really there to try and force back into the cells what's missing. So that's when you get 2 bad copies from mum and dad. Now, my research focuses on eye diseases where you actually only have to get 1 copy, so even slightly more likely just to get 1 copy. And that's passed on to the children. Indeed, sadly, it would be passed to half the children, because when it splits, half of it is going to go, into half of the family will get that 1 bad copy, either from the mother or the father. And unfortunately, that bad copy dominates over the other good copy that you receive. And I think, I'm scared to say, it's almost 10 years I've been working on this, and I've been trying to develop a therapy. And whilst it looks like a powder, it's no different to a Lemsip or any kind of powder medication, within it, it's actually a really precise pair of molecular scissors. And it has the precision of a fighter pilot or a fighter jet, where it's able to hunt out the gene that it's trying to kill, ultimately, and it's able to lock onto it, and it's able to continually cutting it. So it's a very, what we would refer to as a therapeutic pair of scissors, and very powerful and precise. And by designing it specifically to target the gene that we know the person has got a mistake in, we're able to cut it and continually cut that bad copy. So it's a continuous treatment. It's something that we would envisage you would add in eye drops daily, and it's called gene silencing or gene knockdown. And when we're successful, we see that if we can bring down the bad gene by about 50%, we're able to allow the normal copy to then dominate. And I'll show you at the end some of the results that we're seeing. We only have it in animal studies at the minute, and we're trying to progress that. Another kind of more recent, what everyone refers to as game-changing, is genome editing. And I think some of you may have heard of that, called CRISPR-Cas9 genome editing. It's different to gene replacement. It's certainly different to gene silencing. We don't have to go in and cut a gene. We're actually able to almost like rub out that one mistake or tip-X it out and write back in the correct one. So It's really precise, it's very powerful, it's actually quite easy to do and design, and we were very quick to get the first proof of concept for gene editing, not for retinal disease, but for corneal dystrophies at the front of the eye. It really is a remarkable advance in terms of therapeutics available to us as scientists, and I think it's even more notable that we take it and get it from bacteria. We actually hijack a system that naturally occurs in bacteria. I think as we all sit here today in 2023, we've just come out of a virus pandemic and we're all aware of COVID, I don't think we could underestimate the power of a virus. Well, I think you certainly can't underestimate bacteria. The bacteria that have this CRISPR-Cas9, this really powerful tool that I think will be the future of therapy, those bacteria use that CRISPR-Cas9 to kill viruses. So when a virus like the virus that infected all of us exists and it goes towards, I mean, the bacteria that causes food poisoning, the bacteria that's on your toilet seat, the bacteria that you're touching that's making you unwell. When viruses try to infect that bacteria, as part of their immune system, the bacteria use this CRISPR-Cas9. So as scientists who were studying bacteria, they fell upon it, they recognized its capability, and now we're able to use that CRISPR-Cas9 to try and design it specifically to target genes and retinal inherited diseases. So I think CRISPR-Cas9 will definitely be something that will be a therapeutic of the future. And it's unbelievable for me to sit here today and say that we can rewrite an individual's genetic code by using CRISPR-Cas9. Globally and within the UK, and Retina UK are funding specific projects looking at CRISPR-Cas9 gene editing specifically for retinal diseases, but it's been used for many, many diseases and there's great hope for it. There's intensive research going on, editing various genes, and people are rapidly moving towards quite promising therapies. I think you will hear about more of that today for some of the people that are going to touch specifically on therapies. In terms of where we are and the cry out for therapies and why is it taking so long, I definitely think future studies will focus and will have to focus on more novel ways to increase how much of a gene we can get in or how much of a gene therapy we can get into the eye. You hear people saying the eye is immune privileged and the eye will be more susceptible to therapeutics. I think there's a problem with a lot of the gene therapies reaching the amount of therapy that gets delivered to it or the amount of the drug that we get into the eye. So I definitely think more effective delivery methods will be at the center of the future of research in this area. Also, I think earliness and proactiveness of intervention in what we would term a preventative medicine approach is critical for gene therapies. And with the technological advances that we currently have with sequencing and the price of sequencing and the low cost, I think there's no reason why we're not able to offer nearly everyone some form of genetic diagnostics in terms of sequencing and be able to very early on in life, say in a family, the mother has this disease and please be aware there's screening available and child X, Y, and Z also has it. So keep an eye on them and try, hopefully it will become commonstay that these therapies would be given quite young in life for any of the diseases that worsen with time. And people would be acutely aware of looking out for those patterns in younger members of a family. So in all, the enthusiasm for gene-specific therapies to give the doctors the ability that we could actually try and stop these diseases in their tracks, that enthusiasm is still there. And I believe in the therapies that I'm developing, in the gene therapies that I've got just at the animal model stage. And if I just show you here in this next slide, we start off with a mouse model, and the more red, you can see here, I don't have this in front of me, sorry. If you see here where this mouse eye has got a very red color, that's representative of a lot of the bad gene. And when we treat it in that eye over there on the right-hand side, and we don't treat this eye, topically, day by day, you can see there how the reduction in the color comes right down to this level, which means that we've almost got rid of all of the bad gene in that particular mouse. So we transgenically made that mouse to carry the gene, and we labeled it with a really bright color so that we could image it every day. So we could do live animal imaging of topical drops to work out how often should you take the drops, how much stuff do we need to put into it. You never want to add too much of the drug because there's a potential for that drug to go way off and have off-target effects and cut other things and do damage elsewhere. So that's one specific mouse model that we have. Now, that's a front of the eye disease, that's corneal dystrophy. But we add that every day, and then we do experiments where we do what is a washout period. And whenever we stop adding the drops, believe it or not, it gradually, quickly comes back to that within 2 or 3 days. So it's the sort of thing you would have to add every day. In comparison to that last one, which was siRNA, this particular mouse was treated in the right-hand eye with CRISPR. And this was one of those days where it was hot, we were in an animal house, it was extremely smelly, it was very uncomfortable, and it was like, oh, will this CRISPR work? We developed it literally within a week. We added the treatment and we imaged the mice the following morning, and that's what the eye looked like. It completely took it out. So it went straight to the DNA, it cut the bad copy, and that mouse could no longer make that bad copy. And to me, this is like world-changing. We never, ever thought we'd be able to do just a one-off treatment. So this is the CRISPR gene editing. So I think it's why I'm enthusiastic and why other people are enthusiastic about things will change and things should get easier and treatment should be able to accelerate. But that's just an example of what I do. There are many other people and others speaking here today that do fantastic work for the retina. That's it. Thank you.
Thank you, Tara.